Hello students, welcome you all to the online learning platform of Durham College, Assam. In this lecture, we will discuss about glycolysis, which is a very important and interesting topic of biological science, and you have allotted this topic in your BSc 3rd sem syllabus as well as in MSc 1st sem syllabus also. So let us start with our discussion. At the very outset, the first question may come to our mind is what is glycolysis, or what is the meaning of the term glycolysis? To answer your question, I can say that the term glycolysis comes from Greek word glykis, that means sweet or sugar, and lysis, that means splitting or breaking down. That means glycolysis is the breaking down of sweet or sugar molecule. Since glucose is mostly available sugar, widely available sugar in our cell, therefore glycolysis is the breaking down of glucose molecule. And here is the definition, that it is the process where a molecule of glucose, that is a 6-carbon compound, is degraded in a series of enzyme catalyzed reactions to yield 2 molecules of pyruvate, which are 3-carbon compound. During these reactions, some of the free energy released from glucose is conserved in the form of ATP and NADH. In simple language, glycolysis can occur in our cell only when the cell gets sugar molecule or glucose through blood circulation after absorption of our digested food. This process is commonly underwent in almost every living organism as a first step of energy generation. Here you can see the last point, the glycolytic breakdown of glucose is the sole source of metabolic energy in some mammalian tissues and cell types. For example, erythrocytes, that means red blood cells, renal medulla, our brain, sperm cells, etc. Likewise, in case of plants also, some plant tissues are modified to store starch, such as potato tubers, and some aquatic plants, for example watercress, derive most of their energy from glycolysis. Many anaerobic microorganisms are entirely dependent on glycolysis for their energy supplement. It means that glycolysis is very important for entire animal kingdom, plant kingdom, as well as microorganisms also. So in this glycolysis reaction, a set of 10 water soluble enzymes catalyze the reactions and carries out the whole pathway. It takes place in cytosol, that means inside our cell, and doesn't require any molecular oxygen. Now let us see glycolysis process as an overview. So the breakdown of the 6-carbon glucose into 2 molecules of the 3-carbon pyruvate occurs in 10 steps. The first 5 steps constitute the preparatory phase, where the energy of ATP is invested, raising the free energy content of the intermediates, and the last 5 steps constitute the payoff phase, where energy gain comes. Now, let us see the phases of glycolysis at a glance. These are the first 5 phases that make up the preparatory phase of glycolysis. Here, 2 ATP molecules are used in the first and third reaction, as you can see in the diagram. And these last 5 phases comprises the payoff phase. Here we can see that 2 ATP molecules are produced in both 7th and 10th reaction, along with 2 NADH molecules in 6th reaction. And finally, we can calculate the net gain of ATP molecules in glycolysis process. Now, let's have a close look in each and every reactions of glycolysis. So first of all, we will focus on the preparatory phase. As already mentioned, it includes 5 reactions or the first 5 phases of glycolysis. This is the first reaction or first step of glycolysis, where we can see that from a glucose molecule, a glucose 6-phosphate molecule is produced with the help of enzyme hexokinase, and 1 ATP molecule is converted into ADP molecule with the help of magnesium ion. So in this first step, glucose is activated for subsequent reactions by its phosphorylation at carbon 6 position to yield glucose 6-phosphate. And here ATP is the phosphorate donor. This irreversible reaction is catalyzed by enzyme hexokinase as I mentioned already. Next, coming to the reaction 2, here we can see that glucose 6-phosphate is converted to fructose 6-phosphate with the help of enzyme phosphohexose isomerase, and here also magnesium ion is necessary. In the reaction 3, phosphofructokinase 1, this is the name of the enzyme, which is shortly also known as PFK1. So this enzyme catalyzes the transfer of a phosphoryl group from ATP to fructose 6-phosphate to yield fructose 1,6-bisphosphate. In reaction 4, the enzyme aldolase cleave fructose 1,6-bisphosphate, which is a 6-carbon compound, into 2 3-carbon compound, namely dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So this is the reaction 5. This reaction is known as interconversion of the triose phosphates. So here triose phosphates means that this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, both the molecule contain 3 carbon atoms, and that is why they are known as triose molecule, and they contain phosphate group. That is why these two molecules are referred here as triose phosphates. So in this reaction, this dihydroxyacetone phosphate is readily and reversibly converted to glyceraldehyde 3-phosphate by enzyme triose phosphate isomerase, as we can see in blue letters. So this reaction completes the preparatory phase of glycolysis. Next, coming to the payoff phase of glycolysis, which yields ATP and NADH. This is reaction 6 or step 6 of glycolysis. This reaction is the first step in payoff phase, where oxidation of glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate is carried out, and obviously it is carried out by an enzyme which is named as glyceraldehyde 3-phosphate dehydrogenase, and this reaction is also known as oxidation reaction. In the reaction 7 or step 7 of glycolysis, the enzyme phosphoglycerate kinase transfers the high energy phosphoryl group from carboxyl group of 1,3-bisphosphoglycerate to ADP and forming ATP and 3-phosphoglycerate. In this reaction 8, we can see that from 3-phosphoglycerate, 2-phosphoglycerate is formed. So the enzyme phosphoglycerate mutase catalyze a reversible shift of the phosphoryl group between carbon 2 and carbon 3 of glycerate, and here magnesium ion is essential to convert this shift. Now in reaction 8, 2-phosphoglycerate is converted to phosphoenol pyruvate, which is catalyzed by an enzyme enolase, and in this reaction enzyme enolase promotes the removal of water molecule from 2-phosphoglycerate, and finally phosphoenol pyruvate is produced. So this is a dehydration reaction. And finally this is the reaction 10. In this final reaction, transfer of the phosphoryl group from phosphoenol pyruvate to ADP is done. This reaction is catalyzed by enzyme pyruvate kinase, which also requires potassium ion and either magnesium ion or manganese ions. Now we can go for net yield of glycolysis. Here is the overall glycolytic pathway diagram. Instead you can say it as a flow chart. So far we have discussed all the reactions of glycolysis. To summarize the whole reaction pathway, we have to focus on the net yield of energy or ATP molecules. Overall, a total of 4 ATP molecules are produced in reaction 7 and reaction 10 of the payoff phase. However, 2 ATP molecules are utilized in reaction 1 and reaction 3 of the preparatory phase. Therefore, the net gain is 2 molecules of ATP from one round of reaction. The overall reaction of glycolysis shows that net yield of the reaction is 2 ATP molecules, 2 pyruvate and 2 NADH molecule per glucose molecule. Though it is not a highly efficient energy harnessing mechanism, the energy is trapped in 2 pyruvates produced as the end product and 2 NADH molecule. This trapped energy is harnessed through oxidative phosphorylation in mitochondria by using specific machinery for it, that is the cellular respiration process. So the energy generation in mitochondria is started during glycolysis. This energy is stored in mitochondria and released at the time of energy requirement by our cell. So we can conclude that glycolysis is key to cellular respiration pathway. With this, I would like to conclude my lecture with an hope that you have understood the topic. So in our next class, we will discuss about the regulatory mechanisms or regulation of the glycolysis process. So these are some suggested books for you, which you can use for further study. And also I have to mention that all these books are utilized in preparation of this particular lecture. Thank you very much.